This is a quick explanation of the dry cell lab that we're going to do with the Pupp kids today. This is a finalized setup that you can see here. It's fairly straightforward, fairly robust. Will take a lot of errors in fabrication and still give the kids some meaningful data. So remember, there is no right or wrong answer, and hopefully they're going to learn about series and parallel circuits by actually seeing the results without necessarily predicting them. The materials each group will need are seen here: pre-cut pieces of foil and paper towels, some chromatography paper, salt solution with a pipette, a voltmeter and leads. We don't have enough voltmeters for all 8 groups, so we might only do 7 groups. Some alligator leads, some charcoal powder that we will have pre-measured, so they just pick up a little dish of it, and a small electric motor and a paper clip. First step is real straightforward. They're going to take the saturated salt solution. We might see some salt in the bottom of the container. That's fine because it's a saturated solution. They're going to use a pipette and just wet the piece of chromatography paper, which is essentially filter paper. They're going to do that on top of a stack of paper towels. We want it to be super moist, and then they're going to pick it up and just center it on the piece of foil, as seen here. The next step is to prepare the electrode within the dry cell. So the directions ask you to free the alligator clip, and what they're talking about is to move this plastic cover here back down the wire so that the alligator clip itself is exposed. Then you center that on the wet piece of paper, and then you just add the charcoal in a big mound, trying to make sure that the alligator clip stays in contact with the paper. We also want the charcoal to be more in a rectangular shape, bad lines, but we don't want it to be very round, because in the next step we're going to fold this up, and we want to make sure that we can keep the aluminum foil from touching itself. You'll see that in a second. So next, the directions ask the kids to make a burrito. I found that it was better for me to take a ruler, and it's hard to see, but I made marks where I divided the chromatography paper in thirds and not the foil. So then when I did my first fold, I made sure that the foil wasn't touching the foil, and the chromatography paper folded over onto itself. So you did your one fold this way, then you do your second fold this way, and then you do your third fold this way, just as in the directions in the lab. The next step is to connect the voltmeter to the dry cell. The red alligator lead is already inside the burrito. The black one clips onto the outside, and then we connect the alligator clips to the leads from the voltmeter, and there are 2 different kinds. 2 have these little pointy things, and others look like the alligator clips, but the alligator clips can connect to each other. That's not a big deal. The black lead goes into the ground on the voltmeter, and we have 2 different voltmeters, some small gray ones and some larger yellow ones. They do the same thing, so it doesn't matter which one is which. And then you connect everything, and you can get some readings. They both have dials that allow you to switch between measuring current and measuring voltage, and we'll see how much our kids know about what the difference is between those 2 parameters. I'd like to let the kids play with adjusting the scales. It's hard to see on this picture. I should have blown it up, but on the voltage side here, they go from millivolts to volts. So as you change the dial, you're going to see the number changing. And what happens here is if you're in the millivolts and volts, you can see that the digits change. Also, you'll see some fluctuation between 0.8 to 0.81. So they get the idea that these things are not constant and that there's some inaccuracies in scientific measurements. The directions also ask them to press on the cell. This changes the contact between the chemicals in the device and should give them greater voltages and greater currents. And lastly, the students will need to combine groups to connect their cells in series and parallel, and that's where the paper clip comes from. And the directions in the lab packet are very straightforward in how to connect things. They'll measure voltages and currents in series and parallel with and without compression and fill out that table. And then the last step is to attach a motor. It really doesn't matter if it's black to red, red to black. They will spin in either direction. And to tell if the motor is spinning, you can hear it hum. And the higher pitch, the more rapid the spinning of the motor, so the more current or juice is coming out of the dry cell, so to speak, to spin the motor. So hopefully our kids will get some good experience with some basic electronics so that we can then move into making a motor next week.